this video, we are going to look at how antigens are recognized by our immune system. We are aware that we have two different types of cells, that is the B cells as well as the T cells, that are capable of recognizing antigen molecules. In the B cells identifies antigens in the epitopes, which are normally on the surface of an antigen. Now, the T cells are capable of identifying only protein antigens, but that are processed, that are expressed on the surface of an antigen presenting cells. Whereas in the B cells can independently recognize antigens. It does not need the support of antigen presenting cells. This is one of the fundamental difference in the way by which antigens are recognized by in the T and in the B cells. In order to recognize an antigen, there are four important molecules. Which are these four important molecules to recognize an antigen? The first one is a receptor which is present on the B cell. We call that one the B cell receptor. The second is the receptor which is present on the T cells. They are called the T cell receptors. The third is a class of major histocompatibility complex called the class 1 MHC molecules. Then the final one is the class 2 MHC molecules. These are the four different protein complexes that are required to detect or to recognize an antigen. Let us deal with each of these in detail. Let us begin with the B cell receptor. If you look at the B cell receptors, they are very much like the antibody structure. They are specifically speaking, they are antibody itself. The only difference between a secreted antibody and an antibody which is bound on the surface is this will be the structure of a secreted antibody in the Y-shaped structure which we have learned. Whereas the receptor antibodies which are present on the B cells, if this you consider a B cell, if the receptor antibody will be like this, but the difference is this antibody will have a small region, a cytoplasmic, a transmembrane region followed by a very short cytoplasmic region. This the receptor is anchored on the surface of the B cell. What is important to know is this small cytoplasmic tail, that is really small. If this antibody which is expressed on the B cell, if it is of IgM, which is normally the case, IgM and IgD, this cytoplasmic tail is usually about 3 amino acid length. Only 3 amino acid. You can imagine how small that tail is going to be. If it is of IgA, it will be roughly about 14 amino acid length tail, still longer. If it is IgG and IgE, which is expressed on the surface of a cell, it will be about 28 amino acid length. So you can see how this is going to play a crucial role in the signaling mechanism. Now, on every B cell, it is estimated that maybe about 10,000 to maybe about 30,000 such receptors are present on every B cell. And every receptor is capable of binding or identifying only one type of antigenic determinant. Look at the specificity of the B cells. Now, This B cell, this is meant for binding to the antigen. So it can bind to antigen in order to induce an immune response. There has to be many copies of the antigen present, many B cells must be there, and cross-linking must occur. These are all the primary conditions for inducing an immune response. Once it binds, now understand that if the C terminus is very small, it has to communicate a signal to the nucleus of the cell that this is the nature of the antigenic determinant. Construct an antibody which can fit into the antigenic determinant and induce a response or destroy the antigen. That signal has to be communicated. These 3 sequence amino acids are not capable of communicating that because it is too short. Therefore, if the function of this B cell receptor is nothing but binding to antigen. Bind antigen is the function of this receptor. Now that brings us to the next question. If the question is, if B cell receptors are responsible only for binding, who is responsible for the signaling, the communication process to the nucleus of the B cell? Several years of research, especially this kind of research, were carried out in 1980s. So that was a year really immunology developed. So painstaking several researchers found that signal is communicated with the help of a complex, another complex of proteins. And this complex it looks very similar to the immunoglobulin, so it is also called Ig alpha and Ig beta. It is a heterodimer. It's a protein, Ig alpha and Ig beta. Ig alpha and Ig beta, they look very similar to the immunoglobulin structure. Let us represent them here itself. So in the Ig alpha, it will have 2 domains and a transmembrane region, then a long cytoplasmic tail. It has a long cytoplasmic tail because it has to induce a signal, and this has roughly about 61 amino acid. And this is Ig alpha. Next to that is the Ig beta. So Ig beta, they all have disulfide linkages in the domain, a transmembrane region, then a relatively short cytoplasmic tail that has roughly about 48 amino acid in the cytoplasmic tail. And you need such 2 complexes. They will have disulfide linkages connecting, and you need 2 such complexes. Okay, that is Ig beta and Ig alpha is drawn here. So 2 such complexes. This forms a whole complex of B cell receptor. So B cell receptor in association with the Ig alpha and Ig beta, 2 such units, that makes a working or a functional B cell receptor. Now the function of these Ig alpha and Ig beta is, if you look at the detailed structure of these molecules, it has several units that has got tyrosine kinase activity. Tyrosine molecules in these units can be phosphorylated, and this phosphorylation is what will send a signal to the B cell nucleus that this is the antigenic nature of the antigenic determinant. So prepare a kind of an antibody, carry out a gene rearrangement in the genome so that a very specific sequences of amino acid can be made and a very specific antibody can be made against this particular antigen. So this is how a B cell receptor operates.